And welcome to the final lecture in our series on the urinary system. And in part 8 of our lecture series, we're going to take a quick review over what we've covered in the urinary system. Now, kind of to summarize, what we're looking at in terms of the renal function is that blood is going to be carried into the glomerulus, into the glomerular capillary bed of the renal corpuscle. That renal corpuscle, with our epithelial lining, with our fenestrated endothelial cells and our podocytes wrapped around the outside, are going to act as a filter mechanism. And so we're going to allow relatively small materials and fluids to be squeezed out of the bloodstream into the urinary space. And so the other portion of Bowman's capsule, that outer region of Bowman's capsule, is going to kind of collect and funnel our raw filtrate into our proximal convoluted tubules. Our proximal convoluted tubules are going to process that raw filtrate by reabsorbing things like glucose, amino acids, sodium, water, drawing it into the interstitial space where ultimately it's going to return into our blood supply and be circulated in the body. But keep in mind that what we're doing is taking that raw filtrate and, in essence, reclaiming, reabsorbing, recycling materials that get into the raw filtrate that the body wants to maintain, that the body needs. As materials flow from the proximal convoluted tubule, they're going to go into the loop of Henle. And as they flow into the loop of Henle, again, this is the juxtamedullary nephrons, the nephrons that are located close to that boundary between the cortex and the medulla. They're going to have very long limbs. They're going to extend down deep within the medulla, and they're going to establish a hypertonic osmotic gradient. And so, again, the descending limb, because basically the epithelial lining cells are going to be permeable to water and impermeable to salt. We're going to have water drawn out into that interstitium, into the medullary spaces. And in the ascending limb, we're essentially going to have active pumping of salts so that we're permeable to salt, impermeable to water. So we're going to take that high concentration down at the tip of the medulla and essentially come back towards normal as we return our filtrate into the distal convoluted tubule. As the fluid passes into the distal convoluted tubule, we're going to pass by the juxtaglomerular apparatus. And again, keep in mind that the juxtaglomerular apparatus with our juxtaglomerular cells and our macula densa cells are going to be taking a look at blood volume as well as sodium concentration within the urine, and it's going to have the overall effect of releasing factors like renin and aldosterone that adjust the salt, the ion, and the water balance within the body. The urine may be modified a little bit within the distal convoluted tubules, but ultimately is going to pass from the nephron into the collecting ducts, which are then going to pass into the medulla itself. Now, depending upon what's going on within the body, the collecting duct, the principal cells within the collecting tubules and collecting ducts, if they're exposed to ADH, are going to become permeable to water, which is going to allow water to flow from the urine, from the lumen of these vessels, the collecting tubules and the collecting ducts, and essentially flow into the medullary interstitium, that hyperosmotic, that high concentration region within the medulla. And so what happens then is that what is left behind within the collecting tubules and collecting ducts is going to be a reduced volume of hypertonic or reduced volume of very concentrated urine. In the absence of ADH or in the presence of a diuretic factor, what we're going to take a look at is that those medullary collecting ducts are going to remain impermeable to water. And by being impermeable to water, essentially the fluids that are going to be passing from the nephron into the collecting tubules and collecting duct is going to stay unmodified. And so what we're going to do is produce a large volume of urine that's going to be hypotonic, or essentially a large volume of urine, which is at a much lower concentration. That urine then is going to flow into the minor calyx. It's going to be carried by the ureters into the urinary bladder. The urine is going to be temporarily stored within the bladder and then ultimately excreted and released through the urethra. We take a look at the key features of the structures within the kidneys. Our proximal convoluted tubules, we said, were going to be relatively eosinophilic. Simple cuboidal cells with microvilli along their surface, giving it a rough or a brush border type appearance. No clear cell boundaries between the cells because there's a lot of interdigitation, which is going to be present. Fewer nuclei are going to be present there because these are going to be wider cells, and so not every plane of section through the tissue is going to have a nucleus from that cell being present. The distal convoluted tubules are going to be less eosinophilic. They're going to be more classic simple cuboidal, still smooth surfaces along the epithelial lining. Still not as many clear, or still no clear boundaries between the cells because of the interdigitations between adjacent cells, but you're getting to the point where you're going to see more nuclei being present, kind of closer to what you would see normally. The collecting tubules and collecting ducts are going to be lined by our traditional either simple cuboidal or simple columnar epithelium. Nice distinct cells, clear cell boundaries, and virtually every cell profile is going to have a nuclei present. We take a look at the renal corpuscles up there within the cortex. We've got the vascular and urinary pole. The vascular pole, where we've got the afferent arteriole coming into the glomerular capillary bed, the efferent arteriole exiting out of it. Urinary pole, where we're going to be draining Bowman's capsule into the proximal convoluted tubule. That finishes up our lecture series on the urinary system. Please come back for the next series of lectures where we're going to take a look at the endocrine system, the hormone system within the body. As always, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. Thank you.